or no cinema Netflix. So these are the very good example. If you so if you have an idea, you can like make a difference, not for the our country, for the fund. So now I'm coming to the my topic. The my today topic, which is like burden of hepatitis B infection in India and mechanism of mother to baby transmission. So why this hepatitis B we should know about, because this hepatitis B is 100 times infectious than HIV. Everyone know about HIV infection and like worry about, oh, we should have HIV only. But if you know the hepatitis B, this is 100 times infectious and it's leading to the liver cirrhosis, which is not reversible, and it can lead to the hepatocellular carcinoma, which called liver cancer. Yeah. So like whenever you listen the word titus means inflammation, and the hepatitis they means hepatic word used for the liver. So it's called liver inflammation. So if you, there are multiple way where liver injury happen or the liver inflammation happen. It first maybe the immunological damage. It because of something happened to our immune system, and the immune system aggressive give pro-inflammatory responses, and you will get inflammation. Another is infection like hepatitis viruses. The hepatitis virus is A, B, C, D, and E. But among them, hepatitis B is more dangerous because it's leading to a severe infection, and it can cause the liver cancer. So I'm not going to detail on these other like toxic damage, metabolic disorder. So, but this is important. The toxic in India, the major liver problem is happening because of alcohol. So, other than alcohol, it's hepatitis virus. So, if you want to protect your liver, you need to away from the alcohol, and you need to prevent yourself from the hepatitis viruses. So, I'm going the few line about the hepatitis B in the world. Like in, if you say in the like 200 crore people have been infected worldwide from the hepatitis. So that means out of like four, one out of three, one are infected with hepatitis globally. Around 40 crore people chronically infected in India. If you know the Indian population is around 1.35 billion, or like you say 135 crore. Among them 40 crore, more than 40 crore are infected with hepatitis B. Many like the carrier means they don't know they are infected with hepatitis B, but when you they like have some they need when the blood they are giving someone to blood, then they know, oh, I am infected with hepatitis B. So everyone should screen for the hepatitis B, because there is like majority of people are infected in India also. If you see the around 10 to 30 million, that means 1 to 3 crore people infected each year. Everyone, this infection is growing, and estimated 1 million people die each year from the hepatitis B and its complications. So you see that 1 million means 10 lakh people is dying because of hepatitis. So this is a very big issue. And the approximately 2 people die each minute from the hepatitis B infection. So you can see like how this dangerous virus for like humankind. The major cause of cirrhosis and hepatocellular carcinoma worldwide. Now I'm going to the Indian scenario. So if you see the structure of hepatitis B, so this like when you know the like virus, bacteria, or a human, we all have a genetic material. So the hepatitis B is double-stranded DNA, partially double-stranded DNA virus. If you see other than hepatitis B, all viruses are RNA virus for hepatitis, but this is the DNA virus, and this is the HBsAg. Why I'm going to discuss this slide, because when someone will say that give you the result of their blood profile for the liver diseases, you should know after this lecture. So if someone have HBs antigen positivity, this called marker of infection. If someone is positive for HBsAg, you can say he has hepatitis infection. If someone is HBsAg positive and also have HBe antigen positive, that means he is actively like have a high viral load. They will like they need urgent treatment. Okay, so this is marker of replication. That means the person is infected with hepatitis B, and the replication is happening inside their body. And this HBV DNA, The interesting thing is what happened to how this hepatitis B causes cancer. This hepatitis B DNA integrate with our host genomes, and that's why we're not able to eradicate this virus. So for that, how we can prevent it? We need vaccination. So now, like from long, we have hepatitis B vaccination. So we give it like the time of birth. I have one slide for regarding the vaccination also. So, so if you like, we will get the vaccination, then we can prevent from the hepatitis. So another thing, important thing, when you see the liver report, we should see the SGPT or ALT. So if someone have more than 40 ALT or the SGPT, like our liver have injury. That means how we assess that liver have problem on the basis of SGPT or its ALT. So this on the basis of this, if someone have more than 40, that means their liver has injury. Global pattern of chronic hepatitis B. If you see like how we can say the which country is more affected with hepatitis B and like India, what is the status of India? So hepatitis B infection on the basis of the rate, uh, global society have differentiated in three groups: high HBV prevalent country, intermediate, and low. So more than 8% is high. What is the status of India? So that we have this. So the if you see the India is here, so we in India we have 2 to 7% of hepatitis B prevalence or the infection. So India is like intermediate country, but if you the China, Mongolia, Vietnam, all they have red, like they have more than 8% high prevalent. So like the transmission. So when you say like when you say like India have intermediate infection and some country have high prevalence. Why? Like how this transmission happened. So the hepatitis being lightly transmitted through the exposure of body fluids like the HIV. The hepatitis B have the similar route of infection like HIV. And one important thing is vertical transmission. It's called mother to baby vertical transmission. If someone like I am adult or you are adult, then if we get hepatitis B infection, what happens? The 5 to 10% chances we will get chronic infection, which further lead to the cirrhosis and the liver cancer. But what happens if mother are infected like during pregnancy? The baby will get getting chance of hepatitis infection 90%, and their baby will have a liver cancer. Like you must listen like in village or like this cities that baby have born but he has joined this and he died. It's why happened because of hepatitis. Like so now in India we don't have guideline to treat hepatitis during pregnancy, but other country people are giving one drug called tenofovir. If we give tenofovir, we can prevent mother to baby vertical transmission. So this is important. The risk of high 90% if mother is positive for hepatitis B ENT. If mother is ENT positive, what is the E antigen? E antigen is the marker of replication. If mother have high viral load, the virus is replicating inside the mother, the baby will get infection and the baby will die because of liver cancer or liver cirrhosis. So this is very important. So if you want to eradicate the like hepatitis B, we need to take care of pregnant women from these. Now coming to some few important facts about hepatitis B. Hepatitis B prevented with safe and effective vaccines which we have. You can get hepatitis B from the hepatitis B. Cannot get hepatitis B from the hepatitis vaccine. If a person infected with hepatitis B virus does not feel sick, what happened? Like if adult, not everyone have or like symptoms. We become a carrier. How we become a carrier? Like with the corona, its current scenario. Not like everyone don't have symptoms because our immune system is like have a strong immune system, so it's neutralized. They produce like the effective CD8 T cells which clear the virus by the killing them virus infected cell or by the secreting the antibodies against the specific antigens like E antigen for the hepatitis B or the surface antigen. So if you have effective immunity, you will be, like have a virus inside your body, but you will not have symptoms. So like never think that you don't have hepatitis B. Like everyone should have screen in their like So HBV found in blood and other body fluid, and this is same in 100 times more infectious than HIV. So don't worry about HIV. Now think about hepatitis. So if you see here, so like when you like study any disease, you should know how you will detect which part of what sample you will take to test that you have infection. So the high prevalence of hepatitis B, you can like the antigen or the virus you can see in blood and serum. At the moderate level of these virus, you can check in the semen and the vagina and food saliva also. But the low detectable antigen has been found in urine and the sweat and tears and breast milk. So what about the hepatitis B vaccine? So the hepatitis vaccine is against a surface antigen. It's discovered in 1982. The vaccine given intramuscular and usually scheduled at the time of birth as soon as possible within 12 hours of like the delivery, and the 1 month and 6 months. So everyone should get 3 doses. Like sometimes people want like do when they go for delivery, they will get the vaccination and mother like they never visit again for the vaccination. So like take care of it. The vaccine effect will be there only if you gave like the proper at the time. Yeah, so this is the possible outcome of hepatitis B infection. So in this is in case in case of adult, if like the more than 18 year, like I'm not talking about the children or the baby. So some the fate of hepatitis B can be acute infection and the chronic infection. What means acute infection? Acute means that you have got infection from the blood for anything, whatever the possible reason, and within 6 months you will clear the virus. It's called acute infection. Like you will have a symptoms, you have liver injury, everything will be there, but within a 6 month you will clear the virus. So chronic infection means the virus persists for long. If the virus persists long, what happened, it can be a carrier for like 20 year 30 year, and it can go to liver cirrhosis and liver fibrosis and the liver cancer. So in the adult, the chronicity is very less, not that much, 5 to 10%. That means out of 100 people, only 5 to 10 will develop the liver problem, severe problem like cirrhosis and cancer. But if infection happen mother to baby, then what will happen? 90% chance their baby will have liver cancer or cirrhosis. So this is the possible outcome of the hepatitis B infection. So India has around 3 to 4% of foreign carrier. The hepatitis B virus is a hepatotropic DNA virus which is non-cytopathic to the cells. That means what this specific word is very important. Non-cytopathic means the virus did not do anything to our body. Our immune system want to kill the virus. That's why injury happened. So this non-cytopathic virus, but this whatever they induce our immune system, because of this activation of immune system, our body like the liver have injury. So it can be the liver. This hepatitis infection can be acute and chronic, which lead to the cirrhosis, liver cancer. The HBV chronicity is inversely proportional to age. This is very important. If someone like adult infected with hepatitis B, the 5 to 10% chances they will become a severe for the liver diseases. 1 to 5% of baby, only 20 to 30%. If neonates, that means if they got an infection from mother to baby, the 85 to 90% the liver cancer will be there. So the hepatitis B prevalence in India in among the mother pregnant mother is very important, and we studied from Delhi and and here in Madhya Pradesh also, and we found India is the prevalence among hepatitis B pregnant females of hepatitis infection is 1.2 to 1.6. That means around two mother out of 100 are hepatitis B infected, and the mode of hepatitis vertical transmission. So how mother to baby vertical transmission happen? It can be like three ways. First is during pregnancy is called intrauterine infection before delivery. During delivery, that means perinatal because of mixing of blood or rupture of blood the placenta thing, and the transmission after the delivery. So it's because of horizontal transmission, you can say, but before a month or something, for the breast milk, some studies say, but not this is not validated either. So the vertical transmission can be intrauterine, perinatal, and the postnatal. So if like some adult infected with hepatitis B, what they will do? What are the factors influence the pathogenesis or the severity of disease? These are the three factors. First is viral factors, liver pathogenesis, and the immunological factors. So the viral factor are hepatitis B DNA, hepatitis B DNA, HBs antigen, or HBe antigen positivity. So if you give treatment, what happens? You can reach till here, but so far for the hepatitis B, we don't have a cure. If we got chronic infection, after 20 or 30 year also we will have hepatitis. We are not able to eradicate. So for that we need immunotherapy. So we need to understand the immunology of this virus, how we can kill this virus or prevent the injury which occur by this virus. So so we know from the literature and our study that the CD8 dysfunction is major problem. What happened when hepatitis B infection happened? The CD8 T cell non-specific secreting the pro-inflammatory cytokine like TNF, alpha, interferon gamma, all this. What they kill the virus infected cells and the near virus cells. Now our focus is influence the vertical transmission. What are the factor influence the vertical transmission? So we have like published this paper like one of the top journal is maternal viral load and HB antigen status influence vertical transmission. If mother have high viral load and the HB antigen, they will transmit the virus. So what we need to do? If mother like someone is pregnant, we should check hepatitis B status. If she is positive, just we should know about HB antigen. If she is HB antigen positive, we give her treatment as soon as possible because we know that vertical transmission happen and the baby life will be very high. Okay? The high frequency of immature B cell, T regulatory cell, and CD cell is units. What happened? Now, the first is the maternal factor. What about the newborn immunity? Like there are studies which shows the newborn don't have a like mature immune system. So how we can fight? But we have found that the newborn who carry the virus after delivery, they have effective immunity compared to baby who are positive after birth. They have defective immune system. So baby immunity is also playing key role in the vertical transmission. So if you see the overall hepatitis B infection, we have three problems. First, if someone like adult get infection with hepatitis B, he will not convert zero conversion of hepatitis surface antigen. So the surface antigen, which I told in first slide, the marker of infection. So if we have antibody against surface antigen, what will happen? We can prevent the severity of infection. So we don't know how this happened. So we have found one our study. I'm not showing that study now. That one more new cell is called T follicular helper cell. The T follicular helper cell, like a Th2 subset, which help B cell to produce plasma cell, and this plasma cell to secrete antibody. So on the base, if someone have effective T follicular helper cells, they will zero convert hepatitis surface antigen. Second is the vertical transmission, which I'm going to detail, and the role of innate immunity is not clear. Few studies show the innate immunity play major role in hepatitis B, but few say they don't have any clear role. If someone have a like question, you can ask in between also. So if you read in Google or somewhere the hepatitis B, you will know the CCC DNA is called covalently closed circular DNA. The cccDNA, what happened? Why we are not able to eradicate the virus? Because this cccDNA is conserved. If someone like any cell infected, they will keep their copy. So how we will like remove this cccDNA from inside the cell is the problem. And the dysfunction of T cell and insufficient T cell, these are the problem. So the other three target of hepatitis treatment. how we should down the hepatitis B DNA suppression, HBs antigen seroconversion, and HB antigen seroconversion. That means formation of antibody against the replication marker and the infectivity marker. These are the target we have. So we have published these few papers and immune correlate for hepatitis B surface antigen, spontaneous seroconversion hepatitis B. So we found the mechanism of hepatitis B seroconversion, how we can convert it. I'm not going to detail. And we also study the maternal immunity influences the vertical transmission of hepatitis B. This I'm going to show some few slides later. And we also found the placental expression of acyloglycoprotein receptor associated with hepatitis B virus transmission. We found one receptor on the which if that receptor expressed in high level, we can correlate with vertical. We can say this mother will transmit the virus on the basis of that receptor expression. So these are the basic things. This is the very important slide. So how this, like, not about the hepatitis B, other virus, how our immune system fight with it. So when there is any infection, what happens? Our antigen presenting cells, like such as dendritic cell, present antigen to T cell. This T cell differentiate in CD8 T cell and the follicular helper cells. Before like a decade, we know about Th2 cells, this T helper subset 2. But now the recent shows the Th2 among them, the T follicular helper cell a major player who help B cell to become a plasma B cell, which secrete the antibody. And the CD8 T cell kill the virus infection. So if you want to clear any infection, this is like apply for all the viral infection, that we need both immunity. We need the CD8 T cell mediated immunity and we need the B cell mediated immunity. So all together help in the viral clearance and the prevention of disease. So if you see the literature, our question is like one mother is hepatitis B infected, she transmit to virus to their newborn. Other mother are infected, but she is not transmitting the virus to newborn. Why? What is the difference? Why only few mother transmit, not all mother transmit? So how we can make these mother who are transmitting to non-transmitting mother? Our question is that we want to prevent every single children, they should not have a liver disease because they don't have any mistake. Like if their parent did any mistake or happen something, why baby should like wear it? So the key question is, what is the difference of maternal immunity of mother who transmitted the virus to newborn and what do not do so? Why HbA newborn had increased MHOV cells? Why HbA newborn had dismal CD8 cells? So these are the questions which we have studied. So for that, what we have done, we have screened 18,461 pregnant females and we found 1.63% mother were positive. Each positive mother we followed till the delivery and we out this was the 125. 125 mother we have further divided transmitting mother and non-transmitting mother. And we also taken the healthy mother for the comparison. So we studied the immunological response of these mother. What is the difference between these healthy mother, non-transmitting but infected mother and versus infected mothers? What we have done, we have taken the placenta from these mothers and we have done the ASGPR. This is the receptor of hepatitis. Like we never thought about why this like hepatitis virus infect only the liver cells, because they have receptor for the entry, not like all like the corona. Why this effect on lungs only? Because mostly the lungs, because the ACE2 receptor for corona is highly expressed in the lungs, like the hepatitis B. The receptor for hepatitis B is highly expressed in the liver cells. That's why these viruses go and they replicate in the liver cells. Because if someone have receptor, then only virus can go inside the cell. So what we have done, we have done the immunohistochemistry, we have isolated the peripheral blood mononuclear cells, and we have done next generation sequencing or lot of experiment to prove the difference between these methods. So these are the baseline characteristics of these mother. In the clinical way, we didn't find difference in mother who transmitting versus non-transmitting, except the HbA point were higher in mother who are transmitting. We also studied the newborns, and what we found interestingly, this is the first study which shows the receptor of hepatitis B express highly in the mother who are transmitting the virus to their newborn. So if you see this, so this is ISU score for ASGPR is higher in transmitting mother on the mother. So if any mother who express high level of ASGPR, they will transmit the virus. This is the first study which we showed that increased expression of ASGPR and trophoblast cells and HBV transmit. And this we have given this the diagram that how we can prevent from the hepatitis B infection to their newborn if we can decrease the ASGPR expression, so we can prevent the hepatitis B. Now I'm coming to immune mechanism. What happened? This is I'm telling the hepatitis B. This is the antigen. This recognized by TLR cell and the dendritic cells. This dendritic cell show their antigen like presented into T cell. What happened in hepatitis B infection? Like the mother who transmit the virus, they have defective TLR7 expression. So they don't recognize hepatitis B virus. They don't present antigen to T cell. If they don't present T cell to antigen, the T cell differentiation will not happen. Then what will happen? There will be the defective CD4 cell, CD8 T cell, and follicular helper cell. And if there is a defective follicular helper cell, there is a maturation of B cells, and there will be less antibody. There will be impaired antiviral immunity, and overall, what happens? The mother will transmit the virus. If you want to protect your baby, like someone like pregnant, then we need to increase the TLR7 expression. We need to increase the antigen presentation. We need to activate effective T cell differentiation. Then only we can prevent mother to baby vertical transmission. So this is very interesting, and we are publishing in very big journal soon. We found the newborn shows the imprint of mother. So like if like mother have effective immunity, their newborn will have immunity. Mother who did not transmit the virus, what happened? They have effective antigen-presenting cell and they have effective plasma B cell and TFH. The same thing happened to newborns. That means the similar immunity happens in newborn also. In summarize, increased placental SGPR expression, co-localization with HBsAg and so it's role in intra-internal transmission. Newborn shows immune imprint of their mother, and the defective immunity of hepatitis B virus infected pregnant female enhances the chances of hepatitis B vertical transmission. I have not shown the data of next generation sequencing, and the next generation sequencing data also prove the same what I've shown in immunology data, that if the mother have effective T cell signaling related genes, then the newborn also will same. So this is our acknowledgement of lab students, our collaborator, and this work has been done in Institute of Biliary Science, Delhi, and the AIMS Bhopal, few of them. This is the running. So, thank you.
Thank you very much, sir, for such a great informative session on hepatitis B virus and its vertical transmission from mother to the newborn. It is really a great session. I hope all the participants can enjoy the session also and can get a lot of information. So any questions from the participants, from faculty members and from the students who joined the session? Any questions? Anyone can ask the question. I request participants, if they are having any queries, then they can ask the questions. Any students? Anyone? Okay.